Welcome to The Explainer. You know, when you look at a plant or really any botanical medicine, what are you actually seeing? Most of us, we just see a root, a leaf, or maybe just a common kitchen spice. But today, I want you to completely shift your perspective because you're actually looking at a highly advanced, microscopic biochemical factory. Today, we aren't just talking about surface level benefits. We're zooming all the way down to the atomic level. We're going to map the chemical architecture of Curcuma longa, which you probably know as turmeric. We'll put its most fascinating molecules right up on the screen, decode their chemical structures, and reveal exactly how they interact with human cellular receptors to impact everything from severe inflammation to metabolic disorders. So, welcome to the molecular blueprint of Curcuma longa. Here's our quick roadmap for today's explainer. We're starting with the botanical biochemical factory. Then we'll dive into the curcuminoids and their anti-inflammatory core. After that, we explore the terpenes and the volatile oils, map out the cellular targets, and finally, we'll look at overcoming the bioavailability hurdle. Let's get into it. Section 1. The botanical biochemical factory. So, let's set a hard science tone right off the bat here. Curcuma longa is a perennial herb, and its underground rhizome has literally been a cornerstone of ancient medicine for centuries. But from a strictly chemical engineering standpoint, I've got to ask, what actually makes this plant a medicine? I mean, how does a simple soil-dwelling root act as such a pharmacological powerhouse? Well, the answer lies in the plant's rigorous synthesis of secondary metabolites. And honestly, the sheer volume of these metabolites is just staggering. Through some really advanced analytical techniques like mass spectrometry, researchers have successfully isolated and identified roughly 427 distinct chemical compounds from Curcuma species. 427. So we're not just talking about one or two active ingredients here. This is a massive, incredibly diverse phytochemical arsenal. Now, that arsenal is primarily broken down into these major chemical classes. Think of this as the architectural blueprint of the plant. First, we have the famous curcuminoids. These are what give turmeric that brilliant yellow color, and they do the heavy lifting when it comes to anti-inflammatory effects. Then, we have the sesquiterpenes and volatile oils, which give the plant its aromatic properties and antimicrobial defenses. And wrapping it up, we have a wide array of diaryl heptanoids, phenolic compounds, and flavonoids. Basically, each class has a very specific structural motif and a very specific job to do in the body. Moving right along to Section 2, curcuminoids, the anti-inflammatory core. First up is curcumin itself. Chemically speaking, it's a lipophilic polyphenol, specifically diphenyloyl methane. And this guy is the primary heavy lifting worker in our biochemical factory. Now, because it's highly lipophilic, it absolutely loves fat and really struggles in water, which heavily dictates how it moves through the human body. Structurally, its unique scaffold makes it highly pleiotropic, and all that means is it can physically interact with multiple, completely different cellular targets all at the same time. It's quite the multitasker. But here's the crucial point. Curcumin doesn't work alone. It's accompanied by dimethoxycurcumin and bisdimethoxycurcumin. Look at these structurally similar variants for a second. By just slightly altering the methoxy groups on the phenolic rings, the plant creates a suite of molecules that act as natural antagonists against specific human proteins. For example, recent computational and molecular dynamic studies show that these specific curcuminoids act as antagonists to the CLEC2 receptor. Now, why do we care? Because this receptor is involved in tumor cell-induced platelet aggregation. So by binding right there, these molecules actually show the potential to inhibit tumor metastasis pathways. Pretty incredible, right? And this chart brilliantly illustrates the macro effect of all that cellular binding. When macrophages, our immune cells, are challenged with a bacterial toxin, this chemically modified extract steps right in. It significantly suppresses the production of three major pro-inflammatory cytokines, which are basically the alarm bells of the immune system: TNF-alpha, IL-1 beta, and IL-6. So it's not just masking pain. It is actively shutting down the signaling proteins that cause systemic inflammation in the first place. And the effects extend beautifully into metabolic health too. In models of obesity and type 2 diabetes, administering 100 milligrams per kilogram of curcumin significantly impacts the insulin signaling pathway. It actually reduces the activity of two key enzymes that normally block insulin function, PTP1B by 43% and DPP4 by 45%. By inhibiting these specific diabetic targets, curcumin actively improves insulin sensitivity and upregulates glycogen synthesis in the liver. It's literally chemical engineering applied directly to metabolic regulation. All right, Section 3, volatile oils, specifically the terpenes. So, shifting our focus away from the heavy curcuminoids for a minute, let's meet the lighter aromatic fractions. Say hello to R-turmerone. If curcumin is the heavy infantry, R-turmerone is kind of like the volatile scout of the plant. It's a major sesquiterpene that makes up roughly 25.5% of the essential oil of Curcuma longa. This molecule brings its own highly potent antioxidant, neuroprotective, and antimicrobial effects to the table. It has a completely different chemical structure, yet it perfectly complements those heavier polyphenols we just talked about. So why does this matter to you and me? Well, because these volatile components, along with specific isolated peptides from the plant, form a powerful, broad-spectrum defense system. Just look at the data. Turmeric extracts yield a high zone of inhibition against gram-positive bacteria like Staphylococcus aureus, moderate to high inhibition against gram-negative E. coli, and even concentration-dependent inhibition against dangerous fungi like Fusarium solani. It really is a multi-tool against pathogens. It's also super fascinating to look at the botanical genetics here. While Curcuma longa relies heavily on turmerones for its volatile defense, its close botanical cousins, like Curcuma zetawaria and Curcuma erugina, rely on a completely different sesquiterpene called quercetinone. It just goes to show how slight evolutionary shifts in the plant's genome alter the chemical blueprint of the essential oil, totally customizing the plant's defenses. Moving into Section 4, cellular targets and pathways. Okay, we know what the molecules are. Now let's watch them actually work. Take a look at this comparison involving human red blood cells. When our immune system fights pathogens, it produces hypochlorous acid, or HOCl. Now, that's a powerful weapon, but it causes massive collateral oxidative damage to our own cells, literally destroying the normal biconcave shape of red blood cells. But when researchers introduce curcumin, it acts like a molecular shield. Scanning electron microscopy proves that curcumin actually prevents this morphological damage. It scavenges the free radicals and restores the human red blood cells to their normal, healthy biconcave shape. And inside the cell, the mechanics are just incredibly precise. When a bacterial toxin triggers an inflammatory response, it normally sparks a whole genetic chain reaction. But turmeric extracts mechanically intervene. First, they physically wedge themselves between the TLR4 receptor and the MyD88 adapter protein. By breaking this initial link, they completely shut down the downstream IRAK-MAPK assembly line. The ultimate result? A total blockade of the NF-kappa B P65 protein, basically stopping it from entering the cell nucleus and rewriting our DNA to produce inflammation. I mean, it is a highly targeted molecular strike. Finally, Section 5, overcoming the bioavailability hurdle. Now, as amazing as these molecules are in a petri dish, we have to admit they have a major pharmacological flaw. Curcumin is notoriously hydrophobic and rapidly metabolized by the liver, which means very little of it actually reaches your tissues. But to solve this, scientists are turning to modern engineering. One novel physical process is called gun puffing. Sounds crazy, right? But by exposing turmeric to high pressure, water vaporizes instantly, which triggers a Maillard reaction. This literally alters the physical matrix of the plant, creating highly porous structures that massively amplify the release of phenolic compounds and upregulate its antioxidant capacity, all without changing its chemical identity. But even more mind-blowing is what happens when we merge turmeric with nanotechnology. Scientists are now actually carbonizing Curcuma longa to create 17 nanometer carbon dot nanoparticles. By passivating the surface with polyethylene glycol, they transform this highly insoluble turmeric into water-soluble, multicolor fluorescent probes. These nanoparticles are entirely biocompatible, and they're being used right now for advanced, non-toxic cellular imaging. Think about that for a second. We are literally turning ancient spices into microscopic flashlights to study human biology. Which brings us to a really beautiful synthesis of old and new. As researchers Witkin and Lee perfectly stated, ancient wisdom might be built into this interesting and newly appreciated natural molecule. Thousands of years ago, Ayurvedic practitioners knew this rhizome healed the body. Today, thanks to high-performance liquid chromatography and molecular docking studies, we finally have the vocabulary to explain exactly how it does it. So, I want to leave you with this final thought to ponder. If a common yellow powder used to flavor curries actually contains a highly sophisticated, 427 molecule deep pharmacological blueprint capable of altering human cellular pathways, what other molecular blueprints are hiding right now, completely untapped, in your own spice rack? Thank you for exploring this microscopic world with me today. Keep asking questions and keep zooming in.